Yasas from the University of Greifswald, Germany. And today I'm excited to talk about our recent work on detecting phages from diverse environments using machine learning. Phages are the most abundant biological entity on Earth. They display a remarkable diversity in morphology and genetics. Although limited tools once hindered their discovery, the rise of metagenomics has revolutionized our ability to uncover and study them. Metagenomics pipelines analyze microbial communities by extracting, sequencing, and interpreting their genetic content, enabling unbiased discovery of phage diversity. However, many novel phages remain hidden in metagenomic datasets due to current limitations in computational methods. So a recent benchmarking study showed that while many tools now detect phage genome content from assemblies, alignment-based methods offer higher precision and specificity, while alignment-free methods provide greater sensitivity. This led us to ask whether it is possible to develop a more precise alignment-free method approach without sacrificing sensitivity. We developed a deep learning tool with several new features to address the shortcomings of current alignment-free methods. We compiled a more representative dataset, unlike existing methods that train only on prokaryotes by including genomes of other domains of life to better reflect real metagenomic content. Additionally, we developed a novel architecture that captures six-frame that captures distant evolutionary signals similar in spirit to tBLAST-X by translating input sequences in 6 frames during both training and inference. And we integrated a reliability estimation module to detect when the neural network may be making a mistake. So, our model is based on ResNet 18 architecture, but it's customized to handle 6-frame translated nucleotide sequences. The neural network features 2 output heads, a classification head, and a reliability head. Before discussing how this model is trained, I would like to draw your attention to another topic, which is how do we know when a neural network is wrong? So a trustworthy AI system should produce accurate predictions in a known context, and it should refuse to make predictions when the context is unknown. But let's understand what I mean by this. So look at figure A, and it shows the classifier's training data embedding space, and the gray dashed line shows the decision boundary, whereas figure B shows the embedding space of real-world data encountered during deployment of the model. Notice that figure A, the points are packed, and in figure B, the points are a bit dispersed. Moreover, during deployment, the model may misclassify samples from previously unseen classes as one of the classes it was trained on, as you can see here in the red dots. So in our current work, we applied the theory of neural mean discrepancy to address this problem. You can think of it as learning a set of boundary functions around the embedding spaces, and the distance from the boundary function acts as a proxy to the model's confidence in predictions. So let's see how we trained our model. So we first trained the classification model by minimizing categorical cross-entropy. Then we froze the top layer weights to train the reliability head. For training the reliability head, we use the same genome fragments used to train the classifier as the positive class, and we generated the negative class by randomly shuffling these sequences. and then we optimize the model binary cross entropy loss. So at inference time, we slide a window across the input sequence. The neural network then produces class logits and reliability scores for each segment. And then we average these outputs to generate the final prediction. Only if the average reliability score is above a given threshold, we would accept the prediction, otherwise we would reject it. Our neural network captures biological relationships at different levels. As you can see here, instead of one-hot encoding, we learned embeddings that map each amino acid to a dense vector. So shown in top t-SNE plot, amino acids with similar physicochemical properties cluster together. And the bottom t-SNE plot shows the penultimate layers embedding separating sequences by taxonomy. And the figures next to these figures show that the structure, the disappearance of the structure when you shuffle these vectors. Next, to sort of interpret the model's predictions, we generated saliency maps, and we found out that the neural network pays attention to important phage-related genes in both forward and reverse strands. Then we tested our model on simulated metagenomes from soil, gut, and ocean biomes using genomes from species that we excluded during training. And we sourced the biome data from GenBank files. And these simulated data sets are fragmented by generated by fragmenting these genomes using an empirical size distribution we calculated from real metagenome assemblies. We found that Jaeger performs robustly across all tested biomes. In this slide and the slides to follow, the best performing method has a star next to it. So next, we also tested Jaeger on real-world metagenome samples. So panel A here shows the pipeline that we use to generate these real-world data sets. So each sample is first filtered by particle size, separating it to a virus-enriched and a microbe-enriched fraction. And as expected, the viral enriched fraction will contain more detectable viruses. So we actually prepared 3 samples, like 6 samples from 3 biomes. And in the sea water biome, Jaeger reported the fewest predicted viral contigs in the microbial fraction and the second highest number in the viral enriched fraction. In the soil biome, Jaeger outperformed all other methods. And in the human gut biome, Jaeger reported the highest number of viral contigs in the virus enriched fraction, but also the highest number in the microbe enriched fraction. We think this is due to presence of integrated prophages within the microbial genomes in the gut biome. Next, we also benchmarked Jaeger on IMGBR, which contains metagenomically identified viruses. We specifically focused on prokaryotic viruses in this case, and our results show that Jaeger identifies over 97% of the viral sequences in this data set, making it the second best performing tool in our benchmark. Also, it's important to mention that this database was created by the predictions of the top performing method and the 4th best performing method. And panel B shows the runtime comparison of the benchmark tools. And with GPU acceleration, Jaeger is 20 times as fast as the best performing method. This allows for mining huge terabase sized metagenomes in just a few hours without compromising on accuracy. And that's exactly what we did. We ran our method on 17,000 publicly available metagenome assemblies from Magnify and found out that on average about 11% of all contigs in each data set derive from phages. So in summary, Jaeger is a tool for identifying bacteriophage sequences using machine learning. It uses convolutional layers and automatically detect protein level features at 6-frame translation level. And it also effectively handles out-of-distribution samples to reduce false positives. It is easy to install and has a friendly user-friendly interface. Users can also train their own models. They can customize the models, fine-tune the available models, and so on. Jaeger is available on Bioconda as a Python package, Docker, and Aptainer. And we also have a growing user base now. And soon we will also launch a web service for this tool. And I would like to acknowledge Horizon 2020 for funding this research. And I also would like to acknowledge the support I got from all the members of Kadarali Lab and BIO Group. Thank you. Yes, please.
Thank you. That's a very important tool for microbiologists. How well does Jaeger handle outliers like giant viruses, for example, which are, I don't know if they're even phages, are they?
So they are not, so yeah, I think
some of them are
about nucleocytoplasmic large DNA viruses.
Yeah.
Yeah, so our tool will not identify them. They'll just be rejected because they're not phages, obviously.
There are those that do infect bacteria, though, I think, no? Or
no,
am I wrong?
Those,
no? Okay.
Yeah, they infect prokaryotes. Sorry, they infect protists.
Yes.
So, yeah, microbial.
Okay, that was my mistake. Yeah. Well, thank you very much.
Okay, it's fine.
Do you have a sense for how long your kind of input contigs have to be in order for this to work well?
I'm sorry, but I think I can't.
Oh, sorry. Do you have a sense for how long your input contigs need to be in order for this to work well?
So it should work independent of the input size because we are just using a sliding window.
Yeah.
And we don't have any length cutoffs in the program. So it is all based on the final scores that we, like the average score we calculate. But the recent, like the latest, according to the latest studies, phages can be as large as 750,000 base pairs. So.
Sure. Yeah, I was more wondering, like, do you even kind of like have to assemble, like, can you do this from kind of like long short reads or something? Yeah.
Can you please repeat that again?
Sorry, I was wondering if you even have to assemble or if you could also do this starting from something like long short reads.
Yeah, actually, at the moment, I'm trying to extend this method to eukaryotic viruses and other kinds of mobilome elements like plasmids and whatnot. There are a lot of different mobile genetic elements. So that's the next plan, actually. Yeah.
Cool.
Thank you.